So today we're going to cover chapter 8 about transcription. And then next week, or maybe next lecture, I don't think so. Next week we'll do chapter 9. I'm going to have to restructure the quizzes because apparently you're doing the problems at the same time as the quiz, and that's messed up because of the moving the midterm and then trying to reset the clock. Messed up, but it's okay. We'll fix it, okay? So for RNA, it's very simple. It's similar to DNA. There's only a few differences between both molecules. They're both called nucleic acids. The difference is that in RNA, there is the ribose instead of deoxyribose as the sugar. So they still have the same components, which is the phosphate group, as well as the nitrogen base. The ribose, the only difference between ribose and deoxyribose is the fact that there's a hydroxyl group at the 2' carbon, right? And this is why it's called ribose. When you remove the oxygen, it becomes deoxyribose, okay? Second thing, they're both pentose sugars, meaning that they have 5 carbons each, right? So this is important to remember. Second difference is that one of the pyrimidines, which are a group of nitrogen base, is uracil instead of thymine. Thymine. So for RNA, the nucleotides are made up of uracil instead of thymine, and then the difference is only this carbon, this CH3 group right here, okay? Other than that, it's pretty much the same thing. RNA is typically single-stranded, although there are some forms that have double-strand, which is kind of an intermediate structure. It's not the permanent state, okay? So this is what it looks like. It looks like for the nitrogen, for the nucleotides, you still have that phosphate group, which is at the 5' carbon. You still have that nitrogen base that is at the 1' carbon, and then you have this hydrogen group instead of the you have this OH instead of the H at the 2nd carbon, and this makes it the nucleotide for RNA, okay? And this is called uridine 5' monophosphate, and then you have diphosphate, triphosphate, and you have the same thing for arginine, guanine, and cytosine. Okay, this here is for DNA with the thymine and the deoxy here, and so this is just to show you the difference between both. One more difference that they have is the fact that uracil can bind to adenine and to guanine. Okay, not just adenine. So sometimes you'll see uracil bound to guanine, and the chemistry of this is irrelevant for this course, so you don't need to. Because RNA is single-stranded, right? Usually you don't even have to think about this, but what happens is that when you form, for example, a hairpin loop, you'll see U bound to G, and it's just for that specific structure. We'll see that. So the codons are built in a way, so there are codons that make amino acids. That's next lecture or the lecture after. It doesn't really matter which one binds because you'll end up with the same amino acid. We'll see that next chapter, okay? What's important to know is that this is possible here, and it happens in a structure called the hairpin loop, and I'll show you that in a few minutes. Okay. Finally, just to go over the whole structure there. So this is for RNA. You have the same nitrogen bases, the purines, which is adenine and guanine, and the pyrimidines, which is cytosine and uracil. When you attach a ribose to it, right, it becomes adenosine, guanosine, cytidine, and uridine. And then if you add a phosphate, 2 or 3, then you have the mono, triphosphate, okay? These are called nucleosides, as I mentioned last lecture. And these are nucleotides, and the only difference is the phosphate here, okay? Now for RNA, there's different classes that exist. One of the ones that you are most aware of is the messenger RNA, and this is part of the central dogma, which converts DNA to RNA to protein. And so messenger RNA is used by the ribosome to make the protein. There are also different RNA molecules that are used for different functions. So RNA is not just to make proteins, and these are called functional RNAs. And one type of them is transfer RNA, which is used to carry the amino acid to the ribosome. And so when the transfer RNA binds to the mRNA, it detects which amino acid it needs to attach to the polypeptide. Okay, we'll see this all in the next chapter. Okay, small interfering RNAs and microRNAs are used to silence genes. So when you have a messenger RNA built, you can use siRNAs to silence that gene. And what it does is that it binds to the messenger RNA and gets it degraded. And we use this a lot in the lab to knock down genes, okay, by decreasing the amount of messenger RNA for that gene in the cytosol. The others are less known. Ribosomal RNA is important for the ribosome. We'll see how, why is that? But the rest are less known, some are still new and being discovered, right? There's one more type of RNA, is the non-protein coding RNA. So these are just stretches of RNA that have no function up to now. We don't know what they do, okay? So this is what I just talked about. Right, so there's one more thing I didn't mention, is that RNA can have catalytic activity and function as enzyme, and we'll see this. The other types are explained here. Small nuclear RNA are involved in splicing of mRNA, so cutting between the exons. We'll see that in a second. Small nucleolar RNA, first time I hear about them, right? So this is probably something really, really new, and it's involved in processing the ribosomal RNA. And these I talked about, right? So one thing that's interesting, the PV interacting RNAs are, so males have only one X chromosome, female have 2. So if you have 2, that means technically you have double the amount of protein, but that could be dangerous. So there's a mechanism to suppress one of the X chromosomes, right? And it's done through the PV interacting RNAs, okay? And if you remember also, the RNA, so the RNA that's used in the telomerase is made by mRNAs, microRNAs. Okay, it's the same. It's one of the groups. Okay, yeah. So telomerase is an enzyme that has inside an RNA molecule. Telomerase, miRNA, mRNA, microRNA. No problem. Okay, so just like DNA, RNA has directionality, so it's built in a 5' to 3' direction. It's also translated using the ribosome from a 5' to 3', so everything happens 5' to 3'. Okay, that's the easiest way to remember it. Transcription, translation, and replication all happen in a 5' to 3' direction. So now, conventions. This is where I might get you mixed up, but it's important to focus on these conventions because each book says it in a different way, and so you might get mixed up by studying online, okay? By Googling stuff. The convention is that the DNA strand here, the top one, right? And the RNA are written in a 5' to 3'. So this top strand here is called the coding strand because its sequence will be the same as the RNA and will lead to the protein being built. So this is what dictates the protein sequence. The strand that's used to produce the RNA is the secondary strand of the DNA called the template strand. So this is the template to produce the RNA, which is pretty much the same as the coding, right? Does that make sense? Okay. And then what? And then this RNA molecule here in red will be used to produce the protein. And if you see, they're the same sequence, the coding and the RNA. Right? The only difference is the U is replacing the T, but the rest is the exact same. So if you get a problem in the exam or in the quiz or whatever asking you about the RNA, just take the template. Automatically, the other one is the coding, which is the same as the RNA. Okay? Don't start rebuilding it by yourself. It's a waste of time. And then obviously don't forget to replace the T's by the U. What else? I think this is what I just explained. Now in the books and in scientific papers and in other stuff, you'll find different terminology, okay? So I put it all here. Template strand is also known as the non-coding, nonsense, antisense, okay? And I think there was something here I removed. Oh yeah, it's also called crick, but forget about that, okay? The other one, the coding, is the sense strand because it gives sense to this here, the protein. It's also called the coding and the Watson non-template strand, okay? So if you're studying online, just make sure you come back here to figure out what the hell is happening, okay? What else? So in the another term, reverse complement, okay? So whenever you're studying, you'll also see this term here, the reverse complement, which is basically just this one here, okay? I did not put it here in the other slide because I wanted to focus on something. For the exams, if you don't see the direction, assume it's 5' to 3', okay? And then you can make the other sequence from there. Even for RNA, if you don't see the 5' to 3', just assume it's 5' to 3' from left to right, okay? Make sense? This is a big problem that you'll see in the future classes. Everyone uses their own way of calling it, okay? So now for RNA, what's special about RNA is that it's not like DNA replication in the sense that it does not go over the whole chromosome. It goes only at specific places on the chromosome for the targeted genes, okay? That's why you'll see multiple transcription bubbles being open, and I'll show you that in a second. But what you have to think of it is that it's a whole sequence of DNA, and it will only go to places where there's a specific gene that has to be transcribed. And transcription happens on both strands. So if you read the DNA from left to right, you will come to let's say gene number 1. It has a start codon, which we'll talk about, and then it can transcribe from there. But if you read it from the top one, the start codon could be at different place, so it can code for different genes depending on which strand it is, okay? You don't get the same gene, same protein from the two strands. Does that make sense? Because you can read it this way or that way, okay? So that's the first thing. And the second thing is that it's not only one bubble happening. Gene 1, for example, will be targeted on this strand, and there will be thousands of RNA polymerases binding and just transcribing. They come one after the other, okay? While transcription, it's a bit different. It happens only once because you only want to duplicate the DNA once, not 1,000 times, okay? And then depending on the direction, you'll get gene 1 here or gene 2 here, 3, etc. Right. So this is exactly the same thing. The RNA that you are making from the DNA template strand is basically going to be built from a 5' to 3' direction. And this is, each nucleotide will be added at the 3' end, okay, which is basically right here. Direction is going this way, right? But this nucleotide is being added here, etc. Okay, and the nucleotide that will be added is just the one that complements it. Okay, so if you have a C here, then the mRNA will have a G on it. Okay, the only thing is when you have an A, then it becomes a U on the other side. And I think that's pretty much it for this slide. Okay, make sense? Should be. This is the transcription bubble that I was talking to you about. So depending on which strand you are working with, if you are working on the template strand, it looks like this. So basically you have an RNA polymerase going towards the left. Yeah, towards the left, and it unwinds, and then unwinds the DNA and copies it at the same time or transcribes it, and then it will rewind behind it. And then you also have the non-template strand, which is the other strand that's going in the opposite direction. So this here is going in the normal direction, which is 5' to 3'. This one is going in the opposite. That's why we call it the non-template. Okay? Does that make sense? Isabella? Sure? No? I can see you like, okay. So there's two strands, right? The template strand and non-template, right? So the template strand is the one that's being used to make that gene, okay? For gene 1, let's say. And the other strand that's unused, it's called the non-template. Same thing for gene 2. Gene 2 could be on the other strand, so that one becomes template strand. And the bottom one becomes non-template, okay? Just this. That's what I wanted to say. Forget about the direction and all that. Cool. So this is what I meant by multiple RNA polymerases can transcribe the same gene simultaneously. You have the DNA strand like this, and then what's going to happen is that RNA polymerase will start binding here and then start moving along the DNA and producing this mRNA here. So these are all RNA molecules. Some of them are ribosomal, other mRNAs, etc. This on the left here just shows you how complex it is and what's actually happening inside the nucleus. Okay? So these are all RNAs. For example, here you have one gene. So these are all RNAs for one gene. Question. I don't know how I would have done without students for this question, but good thing you guys are here. So if you analyze mRNA from yeast cells and you find that like Chargaff's rule, T replaced by U, this occurs because what? Anybody? No? Yep. Oh, yep. Exactly. Did you cheat? Because I know I posed the answers too, okay? Yeah, so because mRNA is coming exclusively from one strand of the DNA, then obviously you're going to make the opposite of it as RNA, so Chargaff's rule right there. Cool? Good. Okay, so transcription. There are 3 different steps: initiation, elongation, termination. This is pretty much the same for any process you're going to learn in biology, okay? It just keeps renaming them different stuff, but first you start at the promoter. That's the initiation. RNA polymerase binds there, starts elongating along the gene, and then gets off, okay? So we'll go through each step, and we'll see what's different about them. But before we do that, I have to give you a little bit of background on RNA polymerase. So in prokaryotes, there's only one RNA polymerase, and it's made of 5 subunits. In eukaryotes, there are 3 of them, and the RNA is built in the nucleus. So prokaryotes don't have nucleus. So the RNA replication, DNA replication, RNA transcription, the transcription, and then the translation all happen at the same time. So you have replication happening, but at the same time transcription, and that mRNA coming out will bind ribosomes and translation happens. In prokaryotes, because there's a separation between nucleus and cytosol, the translation only happens in the cytosol. So the mRNA has to be ready and kicked out of the nucleus before being translated. And there's 3 different RNA polymerases, 1, 2, and 3. Again, same thing as DNA polymerase. The only difference is that they target different types of RNAs. Polymerase 1 produces ribosomal RNAs, polymerase 3 produces tRNAs, and polymerase 2 is the one that makes the messenger RNA, okay? And they do not need a primer to initiate. They can bind to the DNA directly. Exactly. So remember primase and DNA replication? Well, it's not here in this case. Question? That's the messenger RNAs. Okay. Yep. Yeah, it means that you don't need to add primers to make the transcription, right? So you don't need primase in this case. Okay. Yeah. No, it's more it's more me talking to myself when I'm making these. Yeah, maybe I'll, okay. Cool. So let me see what number of slide we are at. So I can 23. Okay, I think I'm gonna cut it soon. This way we can do the rest next lecture. Yeah, students messed up all my schedule. It's okay. Okay, so I'm probably gonna cut it soon, like maybe for a few more slides, and then leave the rest for next lecture. And then I'm gonna have to think about the quiz, what we're gonna do about it, because you try to kind of give the students quizzes and all that, but you end up doing what they want, which is removing one quiz, right? So this is what I'm thinking about, because we need to restart the problems first, the week after quiz, right? Right now we're doing both at the same time, right? So we'll see with that. Anyways, where was I? So the RNA polymerase in prokaryotes is made up of 5 subunits, okay? 2 alpha, 2 beta, this W, which is omega, and this sigma subunit, which is not part of the RNA polymerase, okay? So the sigma 70 subunit is the one that binds to a region on the DNA called a promoter, and that recognizes where genes start, okay? There's different types of sigma proteins. They all have a number, so sigma 52, 31, etc. And they all target a specific type of genes, okay? So each subunit kind of recognizes a consensus sequence on the DNA, which I'll show you now. For example, the sigma 70, it binds to housekeeping genes. So these are like necessary genes for the organism, okay? Like these ones are always present, always transcribed and expressed. And this is called RPOD, different name for sigma 70. There's another one, for example, called RPOS, sorry. And this is for the general stress response. So when there's stress in the cell, the sigma subunit will be kind of specific for this. These genes will be activated and will be able to bind the genes for stress response and induce a pathway downstream. Does that make sense? Okay. You don't need to know what each of these subunits do other than the sigma here, okay? The others, just assume that it's just a big bubble, and remember that there's 5 of them. Right, so the sigma subunit and the polymerase, they bind to the promoter, and the promoter looks pretty much like this, okay? So inside the promoter, there's 2 different sequences that we need to remember, not the sequence itself, but their position along the DNA. So you have this is what we call a Tata box, because it's just a repeat of TAT, and it's at -10 of the coding sequence. So your DNA has regions that are for coding sequence and the others that are regulatory, okay? The coding starts, for example, here, and this will be the sequence for the protein that you're trying to make. But the mRNA built from this is not just the coding part. It's also this whole section here, right before, which will be the 5' untranslated region, okay? And this one starts from the end of the promoter, okay? So from the end of the promoter, there's the mRNA that will be built. Part of it will be the 5' untranslated region. Then you get the coding sequence, and then there's also a 3' region, which is not shown here, but just to give you an idea. The coding sequence starts at a codon called the start codon, and it's usually ATG, which codes for methionine, okay? We'll see this in one lecture or 2. Now, the promoter is a regulatory region. It's where the RNA polymerase binds, and the sigma protein. Also, the sigma recognizes this -10, -35 sequence and binds to it. So all these genes are bound by the sigma 70, but there are other sigma proteins that recognize different sequences. Yeah. No, so you have the promoter, you see here, the blue stops here. So this is all the promoter. Yeah, it's a bit misleading, but that's good. Make sense? Yep, 10 bases away from the end. Right? Yep. This here, this is a gap that is included in the mRNA, but not in the protein. So it's called the 5' untranslated region, 5' UTR. Okay? Yeah, exactly. So transcription happens. Yeah. It's okay. No, the 5'. It's part of the mRNA, but not the protein. That's why it's called the 5' UTR region, because there's a gap between the promoter and the start codon of the coding sequence, which basically is useless, and it's called the 5' UTR. Why is it there? That's a very good question in terms of evolution and why. I guess there is some space that needs to be allowed for the structure of the RNA polymerase. I'm not sure. We use it in the lab as an important region to kind of knock down proteins, because what you can do, this is advanced research, but I'll try to explain it, okay? So let's say I'm making a mutation here, okay, in this coding sequence. So then I can affect that protein. The way I can put that protein in the cells is by putting the mRNA for it. So I can just put an mRNA that does not have the 5' UTR, right? That starts exactly here and ends exactly here. And then what I can do is use an siRNA to target the 5' UTR. This way, it will destroy the internal mRNA and then allow for the cell to make my own mRNA, which does not have the 5' UTR. Does that make sense? And then I can play with that. Okay, actually I'm doing this right now. What else? So you can see here that it's not super conserved 100%, but still works. Okay, so this is the consensus sequence, which is the best out of all these together. Okay, the Tata box is very conserved. The other sigma proteins, they will detect a different sequence here. Usually the Tata box stays, but the other one changes. Okay. Yeah, the consensus sequence is like not what I mean by the best, so not to mix you, but because amino acid nucleotides change between organisms, but the consensus sequence is the most conserved among those. Okay, so pretty much this here. Okay, I think this I explained. So the transcription. What the hell happened here? The transcription start site is the +1 site. 9 bases down upstream is the Tata box, and then 35 upstream or 34 upstream is the other box there. And then the the start codon is downstream of that +1. Okay, these words that I'm saying, it's important to remember them because you'll use them for your whole life. Okay, downstream, upstream, etc. Okay, what else? Let's do prokaryotic and then stop. Okay, and then we'll continue the rest next lecture, and we'll see. So transcription stops at a sequence called the terminator sequence. Now there are two, so there's elongation, obviously, where the sigma protein binds the promoter, finds the sequence that is specific for it. There is a movement that starts with the RNA polymerase elongating the RNA, and then the sigma will leave at that point. When it starts, the RNA polymerase starts moving, the sigma leaves, and it will start making this mRNA, okay? After that, and it will rewind behind it the DNA molecule, the double strand, okay? Now at the end, what happens is that there will be termination because you can't just produce a transcript nonstop, right? And the way this happens is through a hairpin loop. So you'll see at the end that there is this sequence here. At the end, you don't need to remember it by heart, but this sequence is basically the same on both sides, not complementary, right? And so when the RNA comes out, it will bind to itself, right? And this is called a hairpin loop, and this is what terminates transcription. Yeah. Exact. Most mRNAs have this here. Okay. There's a second mechanism that I'll show you now, but remember earlier I was telling you that you can bind G. I think I put it in the other one. Where is it? Okay, I'll figure this out next lecture. For now, all you need to remember is that there are two types of terminators in prokaryotes. One called the intrinsic. This is usually the main one, and it's also called row independent. And the mechanism is through these hairpins here, right? And then at the end of this sequence, there's a lot of U's together, okay, which will serve later on for the poly A tail, but we'll get to that, okay? So just remember that there is a hairpin loop that gets formed because the sequence allows for it. This sequence is basically this, which is complementary to this, okay? And it will lead to this hairpin loop, which will remove the polymerase from its DNA. The other one is called row dependent. There's this hexamer called a row protein. So basically 6 copies of the same subunit, and they will detect a sequence that is rich in C, but not G. Because it's single-stranded, it makes sense, right? Detect only sequence that has a C, and then it has a helicase activity. So what it will do is that it will follow RNA polymerase and kick it out from here, okay? In this case here, you don't have this poly U sequence at the end of the mRNA, okay? And you don't have this either. Yeah. The hairpin? There's 2 mechanisms, one that does not require row and one that requires row. This one here? Because it will, just the structure of it will kind of mess up the position of the RNA, but also this will be digested. So there will be proteins that will come and destroy this, okay? This one here will just follow the RNA polymerase, and it goes faster than it because it has a helicase function, and it will kick it out. Crazy, this RNA polymerase there. Okay, what else? So bacteria have genes that occur in what we call operons. So you'll see this in like 2 lectures, I think, 3, something like this. But the lac operon, the gal operon, etc. These are also called polycistronic structures. And it's just basically saying that there are mRNAs for this operon that will be built. So usually what you have is only one gene being transcribed, right? But in this case, you'll have multiple genes being transcribed and the same mRNA molecule used to produce different genes, okay, that are part of the same operon. For example, the lac operon will have all its gene on the same RNA molecule, and they will be transcribed from that same RNA molecule instead of 3 separate molecules, okay? What else? And each gene has its own start and stop codon for translation. We'll see this later on. Question. So in which aspect is RNA synthesis not similar to DNA replication? Yep. RNA does not, exact. So we just said this, remember primase, we don't have a primase here in this case. So RNA synthesis does not require an existing primer associated with the DNA template to which nucleotides are added. The rest, same. What else? Okay, I think we'll stop here. Next lecture, we'll be looking at transcription in eukaryotes and at mRNA, what do you call it? Modifications there, like poly A tail and capping and all that. I'll also probably go over the midterm because we have only a few slides in this chapter left. And if you have questions, then we can meet and go over it. Okay, this way it will avoid me meeting 80 students in the office. So I'll go over the questions here quickly. And then if you have more questions about that, we can meet in the office. If next time there's something that actually next time I'll be outside. I'll wait for you guys if you guys want to come. Okay, make sense? That's it. See you later, alligator.